The last material that will be on your exam next Monday, and in study guide for chapter 14, that would correspond to the first 8 questions. Okay? So study guides to the chapter on photosynthesis, for DNA, chapter 13, and then the first 8 questions from study guide chapter 14. Format of the exam will be the same, so shouldn't be any surprises. If you're going to go to the Body 3.0 lecture Thursday evening in the Brannigan room, it would help if you could bring your ID, because they've got a new tracking system now where they can just scan your ID and then I will know you were there. If you don't have an ID or you've lost it or misplaced it, they will have little tickets that they can hand out for proof of attendance as well. I think it'll be a good talk. It'll be interesting to hear what she has to say about growing body organs in the lab for potential replacements for things like hearts and livers and things like that. So I think it'll be a good talk.
Do you want us to write a little summary?
No, you don't have to, but pay attention, you know. Any questions? 7 o'clock, I believe. I just sent out a reminder with by email. Okay. Well, we are talking about ultimately in the next few weeks the process of gene expression, and ultimately what we're trying to get you to wrap your head around is how genotype determines phenotype. Genotype is a reflection of what genes are present in your chromosomes and what you obviously inherited from your parents. And phenotypes are the visible traits that are associated with and determined by those genes. So I think a good place to start out in this particular discussion is we use the term gene all the time, but do we really know what that term means? So what would be a good definition of a gene? Okay, it's something that codes for a protein. So this idea of coding information and determining a protein. What does it determine about the protein? Yeah, it really does determine what that primary structure of that protein is going to be. Because remember we said last time that if you alter either the kind of amino acids in a protein or the order in which they occur, then that structure will fold differently. And the way a protein folds determines its function. And really, it's protein function that ultimately determines the outcome, the trait. Okay? Many times, one of the more important class of proteins that determines functions of cells and organisms are enzymes. But sometimes the proteins aren't enzymes. Sometimes they're structural proteins as well. So it's something that contains coding information. What is that something? What's a gene made up of? Where do we find it? Okay. Genes are found within the nucleus. They are part of a chromosome. And what are chromosomes made up of? DNA and protein. Okay? And all that research that we talked about determined which of those 2 elements actually represents the coding sequence. It's DNA, right? So the coding information is basically just a series of nucleotides. How many genes does a chromosome contain? Okay, how many chromosomes are there? Last semester? How many chromosomes do you have? How many do you get from mom? 23. 23 from dad. 46 homologous pairs. So you've got 46 of these things called chromosomes. Okay? And if each chromosome only contained a single gene, you would only have 46 chromosomes. Anybody have any idea of the current estimate of the number of genes that a human has?
20,000.
Yeah, they keep revising it down a little bit. I mean, we used to say, even a few years ago, 100,000, and now we're down to less than 20,000 genes, but certainly more than 46. So chromosomes contain multiple genes. And one of the things we'll talk about is how those genes are organized. And when it comes time for genes to be expressed, for that information to be turned into protein, how does this cell know where a gene is and how to go about the process of transcribing it and then translating that copy of transcription, because those two processes have to occur for gene expression to occur. So gene expression is really explained by that concept that we've referred to a couple times already, the central dogma, which says that DNA is transcribed into an RNA, and then RNA is then translated into protein. Okay, so that's a pretty good place to begin with our understanding of what a gene is. It's a sequence of DNA nucleotides that specifies the primary structure of a polypeptide chain. It really tells the cell how to make that polypeptide. They're made of nucleotides. Proteins are made of amino acids. So one of the questions we need to grapple with in understanding gene expression is, how do we change information encoded into nucleotide sequences into amino acid sequences? Okay, so there's some kind of language conversion that's going to take place in this process. Well, as it turns out, actually, our understanding of the concept of a gene has undergone an evolution, and it's still really undergoing an evolution. One of the earliest concepts of a gene was that a gene was genetic information that specified a particular enzyme. So the concept was one gene, one enzyme. And this was carried out, these experiments were carried out by two researchers named Beadle and Tatum. They describe a little bit in your reading the Beadle and Tatum experiment. I think it's interesting to read that, but I'm not going to get into detail about that right now. But these individuals were working with mold spores and growing these spores on all these different agar plates and working with mutants of the spores that some could grow on the plates and some couldn't. And they carried out a set of experiments that basically said, if you have this mutation, this enzyme's missing. If you have this mutation, this enzyme's missing. And they developed this concept that a gene encoded the genetic information for making an enzyme. Well, a little later on, that concept had to be modified a little bit. And instead of saying one gene, one enzyme, they said one gene encodes one polypeptide. Why did they have to make that little tweak in the definition? What's wrong with the concept of one gene, one enzyme?
Enzymes are like a specific class of protein.
Okay, not all proteins are enzymes. Absolutely. But even regarding enzymes, are all enzymes made of one polypeptide chain? No, there are some enzymes that have quaternary structure. They're made up of multiple polypeptide chains. An analogous situation occurs in hemoglobin. Hemoglobin isn't really an enzyme, it's a transport protein. But the functional hemoglobin protein is made up of two types of polypeptide chains, alpha and beta chains. We're going to need a gene for both of those polypeptide chains. So it really takes two genes to encode hemoglobin, which is one functional protein. Okay? Well, What they later found out also was that not all genes encode proteins. Oops. I'm missing a slide here. Yes, I am. When genes are transcribed, an RNA molecule is formed, right? How many different kinds of RNAs are there? Actually, several, but what's the 3 RNAs that you're most familiar with? mRNA, tRNA, and rRNA. Okay? What is messenger RNA? That's the molecule that encodes the information for the polypeptide. But in order for that information to be translated into a protein, you need these 2 types of RNAs as well. What role does the ribosomal RNA play? What cellular structures do you need for translation to take place? Ribosomes. What are ribosomes made of? Protein and rRNA. Okay, so we've got these RNAs that make up part of the structure, ribosomes, that are needed to make proteins. You may know what the transfer RNA does?
Does it like make the polypeptide chain?
It really does. It grabs amino acids and hooks them together in the proper order. It's really kind of the translator molecule, if you like, that turns that language of the gene nucleotides into the string of amino acids. Okay? So we need all of those. Well, genes encode messenger RNAs. Are there genes for transfer RNAs as well? Yep. Are there genes for ribosomal RNAs? Yep. Okay. So, a relatively recent concept is this idea of one gene, one cistron. What's a cistron? A cistron is a functional RNA molecule. So this would be a cistron, messenger RNA. But so would this. And so would this. Now, what's making this even more complex now is that relatively recently we're finding out that a messenger RNA can actually be rearranged to make more than one protein. That's called alternative splicing. And so our concept of the gene keeps evolving and evolving, and it's much more complicated than originally Beadle and Tatum envisioned with this idea that just one gene encodes one enzyme. It really is much more complicated than that. As it turns out, our list of RNAs is actually incomplete. We'll talk a little bit later on about some additional types of RNA that are produced, each of which are encoded by their own unique genes, besides the big 3 here.
What did you say alternative genes are?
We'll get back and talk about that in a little bit more detail later on, but basically the old concept was that one mRNA could be translated into one polypeptide. But actually there's this process called alternative splicing, which we're really not ready to get into yet. But basically you can rearrange this mRNA after it's made and make different kinds of proteins out of that same RNA by kind of reconfiguring it. That's alternative splicing. It's kind of hard to talk about it now because we haven't talked about mRNA splicing yet. Okay, so that's kind of for the future. Okay, so here's our central dogma. DNA transcribed into RNA. We talked about the characteristics of RNA and the fact that the 4 nucleotides in RNA are A, G, U, and C. For DNA, they're A, G, T, and C. So that's important. and then RNA is translated into protein. What's the advantage of having this kind of complex relationship of DNA going to RNA going to protein? Why not just turn a gene directly into a protein? Why do you have this middleman here? Why do you have this RNA intermediate? Seems to kind of introduce another level of complexity to the process. Is it really needed?
Well, you don't want to send DNA out of the nucleus, right?
Correct, why not? You don't want to send DNA out of the nucleus, why not?
You don't want to risk it being damaged.
You might not want to risk it being damaged. Okay? That would be a very good reason. Okay? Many times the analogy is made comparing DNA to kind of blueprints of a cell that provides the information for how to make a cell. And that's true to a large extent. So when they were building the student center several years ago, some architect planned with college input what the design of that student center was going to look like. And they put together a set of master plans for that, which included all of the structural work, plumbing, wiring, HVAC, you name it, okay? The totality of what that building was going to look like. And then they used those plans and distributed them to contractors, and each contractor went out and built their particular part. Well, would it be very efficient if there were only a single set of plans? No. Then that way you'd have to wait for the plumbing guys to do all their work before you could start work on the electrical. And so copying the plans multiple times, like the cell copies DNA into RNA, gives you economies of efficiency. Well, if there are only a single group of plans, what if a guy took them home and their kids destroyed them? Then the master plans were gone. If you retain that set of master plans in the architect's office in the safe, and somebody calls up and says, we need more plans, what do? They pull them out and they photocopy them. And then they redistribute those plans to everybody as needed. But they keep the master instructions safe, just like the cell keeps the master instructions safe within the nucleus. So there are a number of advantages to having things work in this fashion as indicated by the central dogma. Now, we did talk about some exceptions to the central dogma, most notably the fact that this flow of genetic information, which is typically indicated as being one way, DNA is always copied into RNA, RNA is always copied into protein. There are some exceptions to that, most notably the HIV retrovirus, which can copy RNA into DNA. To my knowledge, there has not ever been any demonstration that protein can be turned into RNA. But again, never say never, okay? One of the things I want you to understand about science is we establish these constructs and these rules, but life is so complex and interesting that oftentimes there are exceptions to our rules. So, here's basically, diagrammatically, what the central dogma looks like. Here we've got a chromosome that consists of several genes, okay? The genes here are indicated in colors blue, yellow, and green. And one of the things that I do want you to understand, and we'll talk about more later on, is there are these stretches of blue right here, which are DNA sequences, but they aren't actually genes. They don't encode an RNA molecule. What's that all about? Well, we'll talk about that a little bit more in detail later on. So let's say we're looking at this particular gene right here. This is going to encode some information for a particular polypeptide chain. This is a very short polypeptide, only consisting of 4 amino acids. Typical polypeptides in the cell have hundreds or thousands of amino acids. And each one will fold up into a unique three-dimensional shape based upon the sequence of its amino acids and how they interact with one another and how their R groups form tertiary structures. And those unique shapes will then have certain jobs in the cell. Maybe it's an enzyme that catalyzes a reaction like the breakdown of hydrogen peroxide in the cell. So here's our DNA molecule, and one of the two strands contains the encoded information for polypeptide chain structure. That strand is called the template strand. And that is what is going to be copied during the process of transcription. What's the complementary strand there for? The complementary strand there is really for replication. Okay? But the information is encoded into only one of these two strands of the double helix. Now, is it always this top strand right here? It might be for this gene, but for this gene, the template strand might be this one down here. So it varies from gene to gene which one actually encodes the information for polypeptide chain structure. The DNA is copied into RNA, in this case a messenger RNA. That process of transcription relies on complementary base pairing, just like DNA replication does. But again, notice that if this is RNA, then adenine will pair with uracil. There's no thymine in the RNA. This particular process happens pretty quickly, and that would most likely imply that there is an enzyme associated with it. Does anybody know the name of the enzyme that copies DNA into RNA? I've heard of it referred to as a transcriptase before. What's the name of the enzyme that copies DNA into more DNA? The main copying enzyme that puts DNA polymerase. Okay, it makes a polymer. So what do you suppose copies DNA into RNA? RNA polymerase. Okay, so that's one of the enzymes that we're going to have to talk about a little bit more. Fortunately, there aren't nearly the number of enzymes involved in transcription as there are in DNA replication. But, in addition to RNA polymerase, there are some other proteins that must be present in order for transcription to occur. And those are called transcription factors. And there's a whole ton of those. And we keep discovering new transcription factors all the time. That's the bad news. The good news for you is I'm not going to dig into all the transcription factors and make you learn all of those the way I want you to know the enzymes that are involved in replication. Okay. So if you understand that this process of turning DNA into RNA requires an enzyme, RNA polymerase, and some additional other proteins, transcription factors, you're pretty good to go there.
Can't they sometimes be like lipids or sterols?
Later on, we're going to talk about regulation of transcription. And that's where we're going to get into some of that. Okay? There are some other molecules that are involved in regulating this process.
Just to clarify, transcription factors are like turn on a gene?
Yeah, and I'll talk about them in just a little bit more in just a second. But yeah, they've got to be present in order for that polymerase enzyme to copy this into this. So in effect, they do. They kind of turn on that gene. Yep. Well, one of the things that Francis Crick of Watson and Crick was actually interested in was how is information encoded into DNA that actually specifies this primary structure of protein? What's the coding system? How does that work? When you were little, did you ever do a simple substitution code with a friend? Where you would say, okay, we're going to take the alphabet, and for our code that only we know, the letter A stands for B, and the letter C stands for D. Okay, and you made this substitution code. And then you could write a secret message if you knew what the substitutions were. Okay? That's not uncommon in, espionage, for example. It's making these substitution codes. Well, Francis Crick started to think about it and thought, okay, I've got this information. This is my encoding information. And there are only 4 nucleotides. But this is the words that this has to encode. How many words do we have in protein? How many amino acids? 20. Okay? So I've got 4 code words that have to specify 20 outcomes. So, can the DNA genetic code just be a simple substitution? A stands for one amino acid, this another, and this another. No. That simple substitution code would only specify how many amino acids? 4. Okay, so if one nucleotide stood for one amino acid, we come up way short. What if there were 2 of these that were taken together that specified the amino acid? How many could we specify then? How many amino acids could we specify if we have 4 things taken 2 at a time? 4 squared. Actually 16. There are 16 different combinations of 4 things taken 2 at a time. Is that enough? We're still short of the 20. What if the code were 3 nucleotides grouped together? How many combinations of 4 things taken 3 at a time do we have? 4 cubed, or 64. Is that enough? Yeah, that's more than enough. In fact, that's too many. What if we had 4 nucleotides specifying the amino acid? How many combinations? That's the math ability. 4 to the 4th power, 256. That's way too many. Okay. So Francis Crick just kind of thought, well, what's the most likely nature of genetic code? A singlet code? No, we've not enough combinations. A doublet code? Still not enough. A triplet code? That'll give us enough combinations with some to spare. A quadruplet code? Way, way too many combinations. And he said to himself, nature is usually economical. And I just can't imagine why nature would evolve in such a way that we would have 256 different code words for 20 amino acids. 64 makes a whole lot more sense. So Francis Crick very early on deduced that the genetic code that it was a triplet code. 3 consecutive nucleotides equals 1 amino acid. Okay? So ACC, that's a particular amino acid. That's another one. That's another one. What do we call these 3 consecutive nucleotides that specify a particular amino acid? That's a codon. If the code is a triplet code, and there are 64 different possible code words, but only 20 amino acids, then some of the code words must code for the same amino acid. Another way of saying that is that the genetic code is redundant. So more than one code word specifies the same amino acid. Now, as it turns out, later research discovered something interesting about the code. It not only contained the information for amino acids, but built into it were some punctuation marks. And basically those punctuation marks said, here's where a gene stops. And so actually built into the genetic code are 3 stop codons. So 61, code for amino acids. And then 3 of those code words code for stop. If there's a stop, is there probably also a start? There is, but it's kind of interesting. The start codon also specifies an amino acid. That amino acid is the amino acid methionine. Another thing that is very, very interesting about the genetic code is that the genetic code appears to be universal. What do we mean by that?
The specific genes cannot be found in the same spot in different people.
Doesn't have to do with the order of genes.
Is it the same for organisms that use DNA as their genetic information?
All organisms that use DNA for their genetic information use the same code words. That's what universality means. So if an AUC stands for, I don't know even what it is off the top of my head. It stands for methionine. AUC stands for methionine in our cells, or in plant cells, or in a fungus, or in a bacteria. That's really important because would you be able to do the experiment you're doing in lab last week with inserting a gene from a jellyfish into a bacteria and then having that gene expressed into a green fluorescent protein if the genetic codes were different in those 2 organisms? No. So the whole idea of genetic engineering is based upon the universality of the genetic code. Well, as it turns out, however, there are some exceptions, particularly to the universality of the code. There are some exceptions in some ciliates, some little single-celled protozoans, and particularly in genes in mitochondria and chloroplast, where the code words specify different amino acids. But that is the exception and not the rule. Finally, the genetic code, for the most part, is non-overlapping. So what does that mean? Let's go back to this slide right here. So this segment of DNA codes for this segment of RNA. When we talk about the word codon, we're talking about RNA molecules. So this is the codon for tryptophan. This is the codon for phenylalanine. This is for glycine, and this is for serine. Okay? I don't expect you to know those code words and what the correspondence is. So it takes how many nucleotides to encode this 4 amino acid protein? 4 times 3 is 12, right? Because these don't overlap. We read them in distinct groups of 3. What if the code were overlapping? Then this would be the information for the amino acid. This would be the information. This would be the information. Okay? That would be an overlapping system. And we would get a lot more encoded information out of this number of nucleotides in terms of numbers of amino acids. Well, there actually is an exception to this rule of a non-overlapping code. And this exception occurs in certain types of virus. Do you remember what I said viruses are made of? A protein code surrounding some genetic information. And that genetic information contains all the genes to make a new virus. Are viruses very big? No. And some viruses are really, really tiny and small. And one of the things that limits them is they're so small that it's hard to encode all of the genetic information in their DNA to make a new virus. So what do? Instead of having a non-overlapping code, they actually have an overlapping code. And that creates some problems when that viral DNA gets into the cell and is then transcribed by the cell's genetic machine. So that's another exception to some of these rules. Okay, so a triplet code, and by its very nature, because it's triplet, it is redundant. For the most part, the code is universal. What does it suggest evolutionarily if the genetic code is universal? Common ancestry? Yeah. That the genetic code evolved early on in cellular life, and then that genetic code was conserved and used over and over again in all the different organisms, suggesting a common ancestry. For the most part, not overlapping. Okay, here is the genetic code showing you the correspondence between the codons and your corresponding amino acids. And here you can easily see in the bracketed areas right here the redundancies that can happen. So for example, look at this particular UCC, okay? UCA, UCG, and UCU. 4 different code words out of the 61. And they all specify the same amino acid series. Here's our 3 stops. Here's our start that also codes for methionine. Here's another set of redundancies here. There's another here, another here. What pattern do you see in the redundancies? Pardon me?
There's similar.
Similar in where? In what way? First 2 nucleotides are similar. So the difference in redundancy is typically in this 3rd nucleotide. That's going to be important later on. Okay. Can you see any advantages to having a redundant code?
There was a mutation that we would have to act.
Okay, what if an error is made during DNA replication? And we stick an incorrect nucleotide in while DNA is replicating. That is a mutation. Okay? But if that mutation occurs in the 3rd nucleotide of a codon, then the RNA that's made will be the same. Okay? Because of the redundancy of the genetic code. So there's some actual built-in protection from mutation because this genetic code is redundant. You may know what this is? It's a tobacco plant, actually, yeah. Why is it glowing? It's genetically modified. It doesn't contain the jellyfish gene. This has actually been genetically modified so it contains a firefly gene for the enzyme luciferase, which is what allows fireflies to glow. So why did they do this? Well, in part they did it because they wanted to try it and see if they could do it. But actually there's some other reasons in biotechnology why you want to do this particular type of experiment. We'll talk about that a little bit more as we dig into what you're going to be doing in lab the next few weeks. Would this be possible if the genetic code weren't universal? No. This is a real nice demonstration of the universality of the genetic code to take genes from one organism, a firefly, insert them into a plant. Yeah, we got to leave, sorry. You can head out. We'll just wrap up here. Start trying to come back.